Hello everyone. In this video, we are going to talk about the topic of cells. We are going to discuss between the similarities and differences of different types of cells. In Cambridge O-Levels Biology 5090, according to the syllabus, there are three different types of cells. So basically, we are going to talk about animal cell, and then we'll discuss plant cell, and we will try to compare both of them with bacterial cell. So what do you need to know is you need to understand that how these cells are similar and how they are different. There are some organelles that are present in all of these cells and some are missing in the cells. So first of all, we are going to talk about the type of cells. In this video, we'll also discuss the size of cells, and then we'll talk about their shape. We'll also talk about some organelles, some things that surround the cells, for example, cell wall, nucleus, and we'll talk about the plasmids. We'll also discuss about plastids. We'll discuss about mitochondria. So basically, all these organelles, we'll discuss them in detail in the next topic, but for today, we are just going to compare different types of cells on the basis of these organelles, whether they are present or not. So then we have ribosomes, then we have centrioles, then there is vacuole, and the last thing on which we are going to differentiate between them is mode of nutrition. We'll also talk about their mode of reproduction. So on the basis of these things, these cells can be differentiated from one another, and you can learn them, memorize them very easily. Okay? So let's start with the very first thing, that is the type of the cell. We do know that there are two types of cells that exist, that are eukaryotic and then there are prokaryotic. So eukaryotic cells are the ones that have membrane-bounded organelles, whereas prokaryotic cells are not having membrane-bounded organelles. So cell membrane is basically missing overall, and it's also missing on the organelles. Let's talk about their size. Animal cells usually exist in 10 to 100 micrometers, whereas plant cells, they also exist in 10 to 100 micrometers, whereas bacterial cells, they are prokaryotes and they are very tiny and cannot be seen through the microscope. So we have to use the microscope for them, and they are about 0.2 to 2 micrometers. Okay. So let's discuss about their cell structures, how the shape of cells looks like. Okay, in the CAIE examination, they do ask you to draw the cells and then compare them. So when we are supposed to draw the cells, so you should know how does they look like from the outside. You can see that the bacterial cell are having different shapes. They are either bacilli, rod shape, they are either spiral, either vibrio, or cocci. So there are basically four types of bacterial cells. A general bacterial cell outline is shown in the image. Now let's talk about their cell wall. Animal cells, they do not have cell wall, whereas plant cells, they do have a cell wall, and bacterial cell are also having a cell wall. When we talk about the cell wall of plant cell, it is made up of cellulose. And when we talk about the cell wall of plant cell, it is made up of peptidoglycan. We are not discussing here fungi, whereas in fungi, the cell wall is made up of chitin. Let's talk about their nucleus. There is a very prominent nucleus. When we made the slides of animal and plant cell, nucleus can clearly be seen in our microscope. Then if we talk about the nucleus of bacteria, bacteria is actually not having a prominent nucleus. It is having a plasmid and it is having a single-stranded DNA, as you can see in the diagram. The plasmid is used for genetic engineering, and we will discuss about it in the later chapters. So plasmid is having one of its very important in our genetic engineering and biotechnology. Okay, so let's talk about the next thing, that is going to be plastids. So plastids are again very important organelle, and they are found in as we know that organelles, well, membrane bounded organelles, they are present in the plant cells, but where they're absent in the bacterial cell, and plastids are particularly plant organelles, so they are not found in animals as well. They are used to give coloration to the plants, so there is no role of them in animals. Let's talk about mitochondria. It is the powerhouse of the cell. So mitochondria is required by all the organelles or the cells, and it is present in animal cell, in plant cell, but in bacterial cell, actually mitochondria does not exist in its original form, though it is said that mitochondria has been the first organelle, and it looked like the structure of a bacteria. So bacteria itself is mitochondria, you can say that. Let's talk about the ribosomes, the protein factories. They are present in animal cell, they are present in plant cell, they are also present in the bacterial cell because bacteria are also making the proteins. Let's talk about the centrioles. They are existing in all them. We talk about a cell sap vacuole. So cell sap vacuole is basically existing in plant cell. It is not there in animal cell, but animal cell does have very small number of vacuoles, okay? So the function is the same, and then in bacterial cell there is a vacuole, but it is larger than the plant animal cell, okay? Let's talk about the mode of nutrition. This is one of the very important concept. Plants are autotrophic because they perform photosynthesis. They can prepare their own food, whereas animal cells and the bacterial cells, animal cells are heterotrophs. Animal cells, they take the food, whereas plant cells, they are both heterotroph and autotrophs. Even they are called as saprotrophic, the ones which release their enzymes out and takes the nutrients from outside. Then we'll talk about the respiration. Plant cells can do aerobic respiration, animal cells can do also aerobic respiration, whereas bacterial cells can do both. Talk about their reproduction. Plants can do sexual reproduction in higher and asexual production in lower plants. There is binary fission, that is a type of mitosis, and it is seen in the bacterial cells, and this is responsible for growing a lot of bacteria in no time. And then we have plant cells which reproduce by sexual reproduction. We have seen plants reproducing by sexual reproduction in our surroundings, right? Pollination is done, and asexual reproduction is also seen outside. After talking about this, in the topic of cells, there is another objective that you need to cover, is that how can we observe the cells under a microscope. You can prepare the microscopic slides and then they can be observed. We can prepare microscopic slide of onion and cheek cell. In the exam paper, they will ask you to compare the external structure of onion cell, cheek cell, any plant or any animal cell, and you should know any temporary staining technique such as methylene blue or iodine. So this is the objective that you need to cover under this topic. We will be using either methylene blue or iodine technique in our labs, and then we will be performing the practical. In the exam, they won't ask you to perform the practical, but they do ask you to analyze the practical. So for that, you should know how does the structure look like and what's the procedure. First of all, we have to take the epidermis, we have to prepare the slides. The slides will be observed in the microscope, and you can clearly see that the microscopic slides will having regular shaped cells, whereas if we make a cheek cell slide by the same process, the cheek cells won't be having a regular shape, they will be having an irregular shape, and you can see there is a distance between them. So you need to compare them in the exam on the basis of their shape. You have to compare them, whatever is visible in it. Cell wall is very visible in the plant cell, whereas it is absent in the animal cell. You can see the nucleus in the side on the plant onion cell slide, whereas in the cheek cell, the nucleus is in the center. Then people should know how to calculate the magnification. The formula for magnification is the image size divided by the actual size. You have to basically find out the magnification by using this formula in the examination. You should know that in microscope, the objective lens is multiplied with the power of the objective lens is multiplied with the power of the eyepiece to calculate the magnification. And Thank you so much. If you have any questions, you can ask in the comment section.